Welcome to Monobiology. In this lesson, we're going to learn about the virus life cycle. You should have printed off this sheet, or I should have given you this sheet that has the 6 steps on a printed off page. And what I want you to do is I want you to take notes on the page as we go through. There's a couple of things we're going to label here on this particular part. Some of these things are already labeled, as you can see, but we're going to label a couple of things. First of all, this organism right here, actually it's not an organism because it's not made up of cells. This virus right here, a specific type of virus called a bacteriophage. See if I can write this neatly enough. Bacterio, and it's all one word, but I'm running out of space. So I'm going to put my phage. It's all one word, bacteriophage. Okay, it's a bacteriophage because it infects bacteria. This is a bacterial cell. My goodness, that's messy. Try that again. Just my E's aren't very good. Anyway, bacteria. That's a bacteria cell. Okay, so a couple of key things. You will have already labeled a bacteriophage from your textbook. And that's that guy right there. These phage receptors, these are glycoproteins that are on the bacterial cell. And you notice how the bacteriophage's legs, aka their tail fibers, are attached to those glycoproteins on the outside of the cell. That's how the virus lands on the cell. And viruses are specific. Okay, so what that means is this particular virus that you can see right here in this picture would not be able to infect our cells because the glycoproteins would be different on our cells. So specific viruses can only attach to specific cells. Okay, so there would be different cells for every species on the planet. Different viruses for every species on the planet. Okay, so step 1 quite literally is, and you can write this down, step 1, the virus attaches to the host cell. This is the host cell. This is the virus. The virus attaches to the host cell. Okay, in step 2, you can see that the bacteriophage is injecting its viral genome. A viral genome, in most cases for viruses, would be DNA. Okay? So step 2 is the virus injects its DNA. Step 3 is a little bit more complicated. Okay? So I want you to pick a color or a highlighter, and what we're going to do is we are going to make that color the viral genome. So notice how I changed it to red. So I'm going to use red. Okay, so in step 3, the virus DNA becomes a part of the host cell DNA, and the host cell begins making virus parts. These are virus parts. Okay? So again, step 3, the viral DNA becomes part of the host cell DNA, and the host cell begins making virus parts. That's step 4. Step 3, sorry. Step 4 isn't really a step at all. It's just basically the cell continues to make more virus parts. That's it. The host cell continues to make virus parts. Now step 5. Notice what's different in this picture. Notice that all of the parts, the virus parts, have become assembled into new viruses. Okay, so step 5 quite literally is the host cell assembles or puts together new viruses. The host cell puts together new viruses. And then step 6, Step 6 is the host cell becomes filled with viruses and bursts open, releasing the viruses. The host cell becomes filled with viruses and bursts open, releasing the viruses. Now this is an oversimplification. Notice how there's only 1, 2, 3, 4 viruses. In reality, there would be millions of viruses. So when one host cell bursts open, it releases millions of viruses, which means millions of cells. Millions of cells can be infected from that point forward. And then, of course, each cell that gets infected, more than millions of other cells can get infected, and that's how viruses spread. Viruses tend to spread very quickly, and if you don't have an immune system, well, they can kill you very quickly. Fortunately for us, we do have an immune system. We'll talk about that later on. And that, in a nutshell, is how a virus attaches to and makes copies of itself within a host cell.